Hi everyone. My talk today is divided into essentially two themes. One is this idea of chemogenetic suppression, which is basically the methods development, essentially, developing chemogenetic perturbation strategies in non-human primates. The second one is to use that technique to make scientific inferences, and especially to go beyond these usual yes-no answers that most causal studies provide, which is an area involved in a behavior or is an area not involved in a behavior. So I hope we can go beyond that and start pushing towards answering how exactly is an area involved and enabling these perturbation data to engage in falsifying competing models. So in the interest of time, I will start with a very brief overview of the scientific question that our lab is interested in. That is, how does the brain solve core object recognition, which is our ability to rapidly categorize objects like the one that I'm showing you here. So as you can tell, this is an image of a dog. And I can rapidly show you many such images, and you can accurately report all of them, what object was presented in any of these images. So we have been studying this in monkeys because they show very similar behavior to humans, and they can also detect these objects. And we can, in addition, do detailed investigations of the neural mechanisms that are implicated. And decades of work in the lab has led to a very good understanding of the lay of the land in terms of the brain circuits, specifically the ventral visual pathway shown here, that are involved in this task. Our goal, however, is to package this information, this knowledge, into concrete image computable predictive computational models that serve as a token of our understanding of this system. Now, but we are far from achieving this, to be very honest, and to get more information about this system, we need to probe the neural circuits in ways that allows us to test the causal nature of the dependencies between such circuits and behavior. So today I will show you one such attempt at probing the macaque V4, and to test whether that is causally involved in visual object recognition. And as I said earlier, the first half of this effort is purely developing a new technique to integrate with brain systems. This technique involves making a craniotomy in the brain, and then, as I'm showing you here, making injections of specific viruses that transfect neurons and delivers this gene, which leads to the expression of a chemogenetic receptor in the neuron that can be transiently turned on to produce, in this case, neural inhibition. So we choose a site in the brain, in this case in V4, and make few injections in the site. So in our case, we made 3 injections. Here I'm showing you the sagittal section view, where the first injection will be made in the deepest layer, and I have done histological examinations to approximate whether the virus actually works and how far it spreads from its injection site. And so we will usually do 3 injections at each site, and then we'll try to cover some part of V4 by making multiple injections in that region. And these injections were made in V4, over which we implanted UTA arrays to assess neural changes. So we also implanted UTA arrays in IT to estimate downstream changes. So this technique is less invasive beyond the initial surgery compared to pharmacological alternatives or, for example, optogenetics, which requires continued invasive access to the brain areas in some form. And also behavioral testing in this case can be compatible to our home case systems, and also it will enable scaling up the data collection in some sense. In this talk, however, I want to simply try and answer 3 questions with respect to the method. First, when we activate the dreads, can we produce suppression, significant suppression at the level of V4? Does the suppression at V4 lead to suppression in IT? And does that produce related deficits in behavior? And on the scientific front, I would like to elaborate on a framework then to use this data to guide the computational model building. Okay, so first let me elaborate our experimental sessions. So once a monkey is brought in the lab, they do 2 types of tasks in block 1. First, they passively fixate while we present images of different objects in their central visual field and record in V4 and IT. Second, they perform a binary object recognition task, which you can see here. So an image will be shown containing an object, and the monkey has to choose from 2 options which one was shown. So here is an image of an object, and there are 2 options, and if you had paid attention, you could tell that this was an image of a bird. So after block 1, I inject saline or CNO, which is an activator of the dreads, intramuscularly, and repeat these blocks. And the same kind of structure of tasks, the passive fixation followed by object discrimination, will happen in each block from block 1 until block 10 and maybe more. So now let's look at changes I observed in V4. I will focus here on the neural data collected during the passive fixation trials only. I'm showing you here how the neural responses change with respect to block 1 after the injection of saline. And this is what happens after the injection of CNO. And as you can notice, there's a significant reduction starting around 40 minutes post CNO injections. And we can look at some examples of firing rates, neuron by neuron, here around 50 minutes post injection. And we can summarize. I can summarize this across 2 monkeys. Each circle corresponds to a neural recording site, and our data also allows us to zoom into each neural site and look at image by image the changes in V4 neural activity. This is evidence of modest but successful suppression of V4 neurons using this technique, and we can get this across multiple neural sites and images. Next, I look at what factors might explain these image by image neural effects. Since I recorded in V4, I could estimate the classical receptive fields of these neurons. So here I'm showing you data from the left hemisphere V4, which encodes visual stimuli in the right hemifield. And an example estimated receptive field looks something like this. So the objects in the image falls in various parts with respect to these receptive fields. They can be completely outside the receptive fields, as I'm showing you here, or they can be completely almost inside the receptive field of the transfected V4 neurons, as I'm showing you here. And it turns out that the neural image by image delta vectors that I just showed you are best explained by these object position variables, especially the overlap of the visual objects with the transfected V4 receptive fields. Now, looking briefly at what happened to IT responses, I here show you that the suppression shown here as -% change at the level of IT increased as a function of the overlap between the transfected V4 receptive fields and the object position, strongly suggesting that those changes were indeed driven by our manipulations of the V4 circuitry. And in fact, if you look at the behavioral data, just to remind you the behavioral task, the monkey saw an image of an object and then had to choose between 2 given choices. And we had 10 objects, so it created 44 possible tasks, and we had around 10,000 images in total that were shown to these 2 monkeys. First, we observed that the time course of the behavioral deficits are similar to changes observed in V4. We can estimate these changes on a task-by-task level, like we did at an image-by-image level for the neural data. We can do this at a task-by-task level for the behavioral data. But most interestingly, just like the IT suppression effects, the behavioral deficits also increased as a function of the overlap between the transfected V4 receptive fields and the object positions. Again, strongly suggesting that these behavioral deficits were indeed driven by our manipulations of the V4 circuitry, and therefore strongly implicating V4 in this behavior and also demonstrating the validity of the method that I was developing. And just to summarize the main results so far, so I have demonstrated 3 things. First, yes, we can get significant suppression, although it was quite modest at the level of V4, and that suppression led to further downstream suppression of IT neurons that are the primary feed-forward target of V4. And that same suppression in V4 also led to commensurate behavioral deficits that we observed when the monkey was behaving in an object recognition task. So all of which combined supports the validity of the method and provides evidence to support a causal role of V4 in core object recognition. Now, as I promised in the beginning, to look more deeply into the role of V4, I will motivate a framework essentially to incorporate such results in guiding the next generation of brain model development. So how do we do this? The first step involved in this process of testing models with this data is to come up with ways to model the effects of perturbation in existing models. One approach could be, we can use the information about the transfected V4 receptive fields to simulate these sort of model scotomas, essentially, and generate the perturbed version of a model. That's one approach. Another approach is to directly look at the data, and I call this approach the empirical fits. So in this approach, I will use some of the data from the V4 perturbation, the V4 neural responses after perturbation, and estimate the parameter changes that needs to be done in the model to explain those measured data. And once I have done that, then I have created a model of the perturbed brain, and I can use the held out data to do the testing. So step 2 for both of these techniques essentially allows us to compare models developed in this way in how well they can predict the held out perturbation data. So if you remember, there were these object position based factors that could predict the measured V4 changes. And just to motivate the framework, I will show you some preliminary results from our empirical fits approach. When we use the CNN's model predictions instead of the image-based factors, we achieve slightly higher and better fits of the perturbed V4 activity. And not only that, given that there are many competing models, essentially modeling activity in V4 and IT and behavior in this artificial neural network framework, we can then start comparing among those models in how well they can explain this perturbed V4 neural responses. This, to me, is the most important point that I want to motivate today, which is I did not need to do more experiments to convince you that V4 is involved in object recognition. However, the causal perturbation data that I've collected now goes beyond that somewhat trivial inference and enables us to discriminate among competing brain models of the ventral stream. That's where I believe the more interesting and the real work in the future will lie, as you can now test these models on how well they explain the IT responses that I've collected with and without perturbation and the behavioral responses that I've collected with and without perturbation. So I'm really excited to carry this forward and in future test this other new predictions from these perturbed models of the system. And I finally want to thank my amazing lab where I had the great opportunity to work in the last 5 years, and all my funding sources to keep the lights on and enable this research.